I'd like to thank Dr. Andropoulos for putting this panel together and for the opportunity to speak today. I'm going to share my screen. So today I'll be talking about clinical anesthetic neurotoxicity. Is there a phenotype? My name is Caleb Ng from Columbia University. I'm funded by AHRQ, but I don't have any other relevant financial relationships. I'm sure many of you are familiar with this warning, which was put out in 2016, warning that repeated or lengthy use of general anesthetics during surgeries or procedures in children younger than 3 years may affect the development of children's brains. If we're trying to answer the question about whether or not there's a phenotype, first, we need to talk a little bit about the literature. If asked to summarize the literature, many people will say that the results are mixed, with some studies finding an effect while others do not. I think the explanation, however, is a little bit more complicated than that. It's not just that the results are mixed. The studies themselves are also very heterogeneous, including where these children are from, what type of procedure they received, what age they were when they were exposed, what the dose of exposure was, what was measured as an outcome, when the evaluation occurred, what the comparison group was, and finally, the result. So I don't think it's necessarily surprising that you're going to find variability in the results of the studies when there's so much variability in the studies themselves, including what outcome measure was actually evaluated. It's also important to understand that the vast majority of the studies use pre-existing data that was collected for other reasons. There's nothing wrong with this. And most of the studies that I have done are analyses of pre-existing data because there are benefits to this method. If you don't need to collect your own prospective data, some of these studies can be performed much more efficiently. Also, larger studies with larger sample sizes can also be done when you have pre-existing data. And this is important specifically when evaluating anesthetic neurotoxicity, because if we believe that anesthesia has an effect, it's likely to be a modest effect, and larger sample sizes are required to evaluate smaller effects. There are, however, also limitations to pre-existing data. And the first is that the outcomes that we use must be available in the data set. And the issue with that is that the outcomes that are available may not be the most sensitive for answering your question. Another limitation is that pre-existing data may lack important baseline clinical covariates, so residual confounding may be a problem, meaning that there may be other factors besides anesthesia that's driving the differences between exposed and unexposed children, but you cannot account for these factors because you lack the necessary data. But is there a way to avoid some of these limitations, but also have larger sample sizes? One method may be using meta-analyses. This is a meta-analysis that we published nearly a decade ago, and at that time, there's less known about the topic, and there are far fewer studies. But in looking at the studies that we included, they looked at very different outcomes. Here you can see that all of the outcomes that were evaluated, learning, behavior, mental disorder diagnoses, severe disability, adaptive behavior, and language are all different from each other. Another important thing to note in the studies that were included in this meta-analysis is that some of them evaluated children with very high levels of comorbid disease, with one study evaluating very premature neonates, and another looking at children who had open heart surgery, where this cohort experienced a 16% mortality after surgery. In sick kids undergoing major surgery, it may be difficult to dissect out the impact of anesthesia from other major clinical factors. Some studies also evaluated children at a very young age, under the age of 2. And the problem with that is that neurodevelopment is difficult to evaluate in very small children, and it also might not be predictive of long-term function when evaluated very early. Since there are now many more studies available, we recently tried to improve on some of the previously published meta-analyses. We're able to be a little bit more strict with our inclusion and exclusion criteria in choosing studies to include. First, we excluded all studies that focused on children with major chronic conditions because, again, looking at children with these major conditions, it may be difficult to determine the impact of the anesthesia. With regard to inclusion criteria, we evaluated children first, just exposed to a single general anesthetic. We looked at studies that use prospective outcome assessment. And we did this because these studies were designed to use the most sensitive outcome measures and also allowed for a review of clinical data in order to minimize confounding. We also looked at studies assessing children who are school-aged or greater because these children may also be more reliably assessed and their outcomes may predict long-term function. After identifying these studies, we evaluated all the primary and secondary outcomes in these studies. We performed meta-analyses on the results from these studies that used the exact same outcome measures. The reason why we did this is because we wanted to have the cleanest study possible. We wanted to ensure that we were comparing apples and apples and not apples and oranges. Overall, 8 studies met the inclusion and exclusion criteria. And of these 8 studies, there are 3 studies that had overlapping outcome measures. The 3 studies included in the primary analysis were the PANDA study, which is a multi-center ambidirectional study where children with anesthesia and surgery were compared to their siblings who had no anesthesia or surgery. The MASK study, which is another ambidirectional study where children with anesthesia and surgery were compared to other children in the community without anesthesia or surgery. These are both observational studies. And the third is the GAST trial, which is unique because it is an international blinded randomized controlled trial. And the first large-scale trial and only large-scale trial evaluating this question. In this study, the researchers randomized children to either get sevoflurane general anesthesia or awake regional anesthesia without any sevoflurane for children undergoing hernia surgery. The primary outcome for all 3 studies was IQ. In combining the data from all 3 studies, we found that there had 5 outcomes in common. The first was intelligence as measured by IQ. And in looking at this outcome, there was no difference in IQ scores with a single exposure. You can see that the point estimates are all around 0, with the 95% confidence intervals all crossing 0. It's also interesting to note that these results are very similar between the 3 studies. Here, you can actually quantify it with an I-square statistic of 0, which means that there's little or no variability between these 3 studies. The next outcomes that we evaluated were the CBCL outcomes, of which there were 3: total behavioral problems, externalizing behavioral problems, such as acting out or aggression, internalizing behavioral problems, such as depression or anxiety. And these behavioral problem scores are a little bit different than the IQ scores in that with IQ, the higher your IQ, the better. However, with behavioral problem scores, the higher the score, the more problems you have, so the worse the score is. And overall, what we found was that with regard to the CBCL scores, there were higher or worse behavioral scores after a single exposure of about 2 points. And that the size of the association was very similar in all 3 studies. Again, the I-squared statistics were 0, showing little or no heterogeneity between the studies. The final outcome was the brief, which evaluated executive function problems. And with regard to this outcome, the children with a single exposure had higher scores by about 2.6 points, so more executive function problems. However, this final outcome was not statistically significant after multiple comparisons adjustment, and it also found significant heterogeneity in this outcome of 69%, meaning that there was significant or serious heterogeneity between the studies. And the reason for that, you can see here, is that the MASK and the GAS study both found worse problem scores in anesthetic exposed children, but the PAN study did not. So one of the tricky things about score differences is to try to figure out what a 2-point difference actually means from a clinical standpoint. To try to help with interpretation, we did a secondary analysis to determine if this 2-point difference was associated with clinical deficit. We defined clinical deficit as a score that was 1 standard deviation worse than the mean score of the population, which also is defined as looking at the percentage of children in the worst 16% of all normalized scores. And these were the results. Overall, we found that there is no statistically significant increased risk of being in the worst 16% of all normalized scores in total behavioral problems and externalizing behavioral problem scores. However, there was a 47% increased risk of being in the deficit range on internalizing behavioral scores, as well as a 68% increased risk of being in the deficit range of executive function scores. One of the problems, or one of the questions rather, that often comes up is why we use 1 standard deviation as a threshold, and why this threshold was used in the MASK and the PANDA studies. Often, to make a diagnosis of a pathologic behavioral deficit, children may need to score 2 standard deviations away from the mean, or in the worst, 2.5% of all scores. And there are 3 reasons why this was not used. The first is sample size. As you may know, the rarer the outcome, the larger the sample size needed to evaluate that outcome. And being in the worst 2.5% is a very rare outcome. The second is that if the population is shifted, which means that, and that appears to be the case, since there is a 2-point mean difference in exposed and unexposed children, it likely will not matter what threshold you choose, as you should theoretically have a similar percentage of children crossing any threshold. And the third reason is that if you only consider a child of clinical deficit if they have scores that are 2 standard deviations worse than the mean, that's a pretty high bar. That's equivalent to saying that you only consider a child to have a deficit in intelligence if they have an IQ score of less than 70, which is 2 standard deviations away from the mean, or a score in the mental retardation range. So the next question that comes up is, what are CBCL in brief? They're validated parent-reported surveys where caregivers will fill out and answer questions describing problems in their child's behavior or executive function. The follow-up question is that if parents know that their kids got general anesthesia, could they be biased in filling out the surveys? And if they are, is the bias the reason for the reported differences? We're able to explore this question using the GAS trial data in another secondary analysis. The GAS trial is very interesting because approximately 50% of parents were blinded to the anesthetic that their child received, so they did not know if their child got a GA or a regional anesthetic. So in the unblinded parents, the children got either a GA or a regional anesthetic. And if there was bias involved, then those parents would likely rate the GA exposed children as having higher or worse scores, and the regional children as having lower or better scores. For the blinded parents, they had no idea what their children received with regard to GA or regional. So if it was bias that was driving this effect, then in the blinded parents who did not know what their children got, they should report no difference in GA or regional. But what did the study show? In the unblinded parents, the GA exposed children were found to have higher or worse scores than the regional anesthetic children. But in the blinded parents, they also reported higher or worse scores in the GA exposed children than the regional children. So in the GAS trial, it seemed that parental knowledge of general anesthetic exposure did not appear to bias the survey outcomes. So to summarize, there appears to be a lot of heterogeneity in the clinical studies, which makes it difficult to interpret and also difficult to identify a phenotype. In looking specifically at the outcomes that were used, it appears that single anesthetic exposure may only be associated with deficits in certain outcomes in certain cognitive domains, and that there don't appear to be any differences in IQ, but there are some behavioral differences reported. There are many other studies that have also reported little or no difference in academic achievement in anesthetic exposed children. However, there are some studies that have reported higher rates of ADHD. So the question is there a phenotype? And the answer is maybe, in my opinion. Well, I don't think that there is enough evidence to change clinical management, since there may be unintended consequences and causality has not been firmly established. I do think that there's enough evidence to continue to explore the question of whether anesthetic exposure causes deficits in specific domains, such as behavior. This is my contact information. Thank you for your attention, and I'm happy to take any questions.